This is part 2 of 5 on Chapter 9, Microbial Genetics. So just to review kind of what we're looking at in this chapter, we're looking at genetics, which is a study of heredity. And in the science of genetics, we're exploring these 4 things. So transmission of the biological trait from parent to offspring, expression and variation of those traits, structure and function of our genetic material, and then how this material changes. In the first part, we looked at the structure and function of the genetic material, looking at DNA and how it comes together and how it replicates. In this section, we're going to be looking at the second part, so expression of these traits, so how this genetic material codes for different proteins that gives us our traits and characteristics. So this is called the central dogma of molecular biology. So this is where we're going to take our instructions in our DNA molecule. Our DNA is going to go through a process called transcription. So this is where the information stored on the DNA molecule is copied into an RNA molecule. So we're going to take information in DNA and then recode it into a messenger RNA molecule called an mRNA molecule. Then that mRNA molecule is going to go through a process called translation. This is where the information contained in the mRNA molecule that we just made from transcription, now we're going to translate that mRNA molecule and use that information to produce different types of proteins. Okay. So this is how we go from DNA. We use the DNA to make an mRNA molecule, and then we use the mRNA molecule to make our protein at the very end. So this is how our genetic information in the DNA is actually expressed in the cell in the form of proteins. So transcription and translation, we're going to first look at the participants. We need RNA, and I already talked about one type of RNA called messenger RNA, or this is called mRNA. There's also 2 other types, transfer RNA or tRNA, and ribosomal RNA or rRNA. So there's 3 types of RNA that we're going to see. And then we also need ribosomes. And hopefully ribosomes sound familiar. We learned about them in one of the previous chapters. So ribosomes are responsible for protein synthesis. And ribosomes are found in both prokaryotic and eukaryotic cells. So the RNAs, RNA is a single-stranded molecule made up of nucleotides. So remember, an RNA is a type of nucleic acid. So RNA, in our nucleotide, you need a sugar. So DNA has the sugar deoxyribose. RNA has the sugar ribose. So they have different sugars. And then also attached to your sugar, you need a nitrogen base. So our nitrogen bases, we have 4 possibilities. So there's adenine, uracil, guanine, and cytosine. So you notice uracil is in blue up here. So RNA has uracil, whereas DNA has thymine. So this is another difference between RNA and DNA. And then in addition to your ribose and one of your 4 nitrogen bases, you need a phosphate group. So again, this is your nucleotide. It's very similar to a DNA nucleotide, except that you have a ribose sugar and one of your possible nitrogen bases is different. So messenger RNA, our first type of RNA, so the mRNA, this is the molecule. It carries your DNA message through a complementary copy. And this message is carried in triplets called codons. And if you look at the picture, we have a bunch of nucleotides in a row making up this RNA molecule. So if you start on the left, we have AUG. So those 3 nucleotides, they make up codon number 1. So you have a triplet, you have 3 nucleotides, and that's your first codon. The next 3 nucleotides, so the CUG in the middle, that's codon number 2. And then your third codon is ACU, right here. So these codons are going to be important later. The second type of RNA is called transfer RNA or tRNA. tRNA, it's made through transcription from a DNA template. And this tRNA, it loops, so we have a secondary structure creates loops. So you can see the images on the bottom here. You have this RNA molecule. It's looping together to form this kind of T or clover leaf structure. So the bottom loop exposes a triplet of nucleotides called an anticodon. So here we have an anticodon. This anticodon, it designates specificity and complements an mRNA. So remember, we just talked about mRNA has codons, tRNA has an anticodon. These codons and anticodons are complementary to each other. So the adenine and the uracil are going to pair up with each other. The guanine and the cytosine will pair up with each other. So that's the complementary base pairing. Okay. So on the bottom of your tRNA, you have the anticodon, and it's in the yellow at the very bottom. It's labeled with anticodon. On the other side of the molecule, the top of the molecule, there's an amino acid attachment site. This is where your amino acid is going to be attached. Remember, amino acids are building blocks for proteins. So these amino acids are going to be put together to make up the protein that we're creating in this process. And then our other type of RNA, the third type, is the ribosomal RNA or the rRNA. An rRNA, it's coded for by DNA, and it's found in a ribosome. So it's a component of ribosomes where the actual protein synthesis occurs. So ribosomal RNA, it's found in the ribosome, and it helps with the translation process. And then finally, we have our ribosomes. So ribosomes are composed of 2 protein subunits, and they're made up of these proteins, and they also have rRNA molecules associated with it. And there's 3 sites on your ribosome up here. And I'll go through them quickly. So we have the A site right here on the right. So first I should point out the ribosome is made up of a large subunit, the big blue subunit on the top, and then a small subunit on the bottom, down here. So on your ribosome you have an A site. This is where your tRNA enters into the ribosome. You have a P site in the middle. This is where the amino acids are hooked together using a peptide bond. And then the third site is an exit site. This is where your tRNA is going to exit out of the ribosome. So it's kind of like a conveyor belt. The tRNA enters in with its amino acid, the green on the top of the tRNA. When we get to the P site, those amino acids get hooked together, and then that tRNA is going to exit out when it gets to the left side of the ribosome. Alright, so we're going to look at transcription first, which is the first stage of gene expression. So this is where your DNA information is going to be turned into an mRNA molecule that holds the information. Okay, so you have a DNA gene, and then you're going to have an enzyme called RNA polymerase. This RNA polymerase enzyme is going to bind to a region on the gene called a promoter. And this promoter is upstream or at the beginning of the gene. So RNA polymerase binds to a promoter, so it's a specific region where this enzyme is going to bind on. Step number 2, the RNA polymerase is going to add RNA nucleotides that are complementary to the template strand of your DNA. An RNA polymerase, it's a polymerase, so it's also only going to go in one direction. It goes 5 to 3 prime. Okay, the third important thing is instead of a thymine, you're going to be putting uracil in because you're making RNA. So remember, RNA has uracil, DNA has thymine. So we're going to be placing a uracil as a complementary to the DNA adenine molecule. And this RNA polymerase travels down your DNA until it gets to a termination sequence. So step number 4, at termination, your RNA polymerase, that enzyme, recognizes the termination signal and releases the transcript. Okay. And your termination sequence is 100 to 1,200 base pairs long. But you have this promoter, the starting where your RNA polymerase binds on, your RNA polymerase travels down, making a complementary strand, and then it reaches a terminator, and your RNA polymerase releases everything. Okay, so this is going to show that step process that we just went through. So, up at the top, we have a red and blue DNA molecule, and one of the strands on your DNA molecule is called the template strand. This is a strand we're going to be using to make our mRNA. The other side of your DNA molecule is called a non-template strand. So on your template strand, there's going to be a promoter region. So this is the starting region. That's where your RNA polymerase is going to attach on. Once your RNA polymerase attaches on to the promoter region, it can start traveling down your template strand. So now we're going to the second image, the second DNA strand in the middle. So the direction of transcription, you're going from left to right. Your RNA polymerase is traveling to the right. As your RNA polymerase travels, it's going to add on mRNA nucleotides to the strand. So you can see our mRNA molecule is the yellow one that's being produced. Your RNA polymerase keeps on traveling down the DNA reading the template strand until it gets to the terminator sequence. Once it gets to the terminator sequence, your RNA polymerase is going to release off, and then you have an mRNA strand that's created, the yellow strand. Alright, once your mRNA is created using this transcription process, in eukaryotic cells, that mRNA is called a pre-mRNA. And in eukaryotic cells, your pre-mRNA has to go through processing. So one type of processing is called splicing. This is where we're going to remove what are parts of your mRNA called introns. And once you remove the introns, you connect the extra parts, which are called exons. So in your DNA template, you have exons and introns. All of your exons and introns are go through transcription to make your pre-mRNA. So that pre-mRNA, we have pink exons and green introns. So we have special proteins called spliceosomes that do the splicing process. The spliceosomes, they are going to cut out the introns, so they're cutting out the green sections of your mRNA molecule, and then they attach the pink sections to each other. And this is called, again, splicing. So we end up with an mRNA molecule that's mature, and it's just made up of these exons, these parts of the RNA that are going to actually be used to make the protein. Other processing that can occur in eukaryotic cells, you can have, besides the splicing that we just went over, you have 5' capping, where on the 5' end of your mRNA, you put a little cap on it. And then on the 3' end on the other side, you have a poly A tail. Basically, you put a bunch of adenine nucleotides. And it can be anywhere from 100 to 200 adenine nucleotides are added on. The 5' capping and the 3' poly A tail, these help with stabilizing your mRNA molecule so that it's going to last a little bit longer. Okay, so just to review, we're interpreting our DNA. So we've gone through transcription. This is where we take our DNA molecule and we use it to make a complementary mRNA molecule. So our DNA molecule is the blue and the pink strands up at the top. You have a template strand and a non-template strand. You use the template strand to make a complementary mRNA molecule. In eukaryotic cells, your mRNA has to go through processing, so the splicing, the 5' cap, and the poly A tail. In prokaryotic cells, these things do not happen. So only eukaryotic cells have to do the processing of the mRNA. So the next thing that occurs is called translation. This is where we're going to take our mRNA molecule and use it to make our protein, put our amino acids together. So during translation, we have our tRNAs. So remember tRNAs? The tRNAs, they use their anticodons to interpret the mRNA codons. So anticodons and codons are complementary to each other. And then on the other end of the tRNA, you have the amino acid. So if you look at this image over here, so on our yellow mRNA strand in the middle, we have these codons. So the first codon, AUG, on the left. This codon is complementary to the tRNA anticodon. So AUG, complementary UAC. You can see that. And then on the other end of your tRNA, you have your amino acid attached. In this case, it's called the F-methionine right here. And then if we go down, we have our second codon, CUG, on the yellow strand. That's the second codon. So the anticodon to that is GAC on our tRNA. And on the other side of our tRNA, you have the amino acid leucine attached to it. And then you can look at the third and the fourth codon. And you can see you have the anticodons that match up with it, and then you have your amino acids on the other end of your tRNA. Okay, so we're going to go through this translation process. And this is the second stage of gene expression. So this is where all the elements needed to synthesize a protein that are brought together on our structure, the ribosome. So we're going to bring in our mRNA, we're going to bring in our tRNAs, we're going to have our amino acids attached to the tRNAs, we have our large subunit and small subunit of the ribosome come together and hold all of this together. And this is where we're going to attach these amino acids to each other to make our protein. And the process that happens is we have to use what's called the master genetic code. And this code is universal among all organisms. And also the second thing is the code is redundant. So first I'm going to show you how to use the chart. So over on the left hand side you see it says first base position over here. So let's say we have the codon AUG. So in the first base position we have an A, so you go in the third box down from the top. And then across the top of this chart it says second base position. So we have A, the second letter in our codon is U. So we go from the left we go into the A box, and then from the top we're in the U box, so the first box. And then in that box you have AUU, AUC, AUA, and AUG. And we're looking for the AUG codon right here. And then that AUG codon, to the right of it, it tells you which amino acid you're going to be attaching, or which amino acid is going to be on that tRNA that we're going to be attaching together. Okay, so let's do another one. How about CAA? So our codon is CAA. So on the left-hand side, we look for C, which is the second one down. Okay. And then in our second position, we have an A. So we go across the top and find A. So we just follow those two boxes together. So you have, in your box, you have CAU, CAC, CAA, CAG. We're in that box. And I said CAA. So look for CAA. That codon tells you that we're going to have a glutamine amino acid attached next. So this is how you use the chart. The other thing, the code is redundant, so if you look at the glutamine that we just found, there are two codons that actually code for it. So some amino acids have two codons that code for them. Other amino acids, like if you go to the box right next to the one we are in to the right, the arginine, you have 4 different codons that can code for that one amino acid. So that's the redundant part of the code. So we're going to be using this. So this translation process we're going to go through, there's 3 steps to it. So you have initiation, you have elongation, and then you have the termination step. So first off, initiation. The ribosome assembles, so the large unit and the subunit of the ribosome are usually separate from each other. So the two parts of the ribosome, they're going to come together on the 5' end of an mRNA molecule, transcript, that we just made through transcription. So our two subunits come together and they hold the mRNA in between them. The ribosome then starts to move along the mRNA and it scans the mRNA until it reaches a start codon. And the start codon is AUG. So always look for AUG first. Don't just start with the first 3 letters on your mRNA, look for AUG and start there, because that's your start codon. Then what happens is your first tRNA molecule with a complementary anticodon. So if our codon is AUG, our anticodon will be UAC. So a tRNA molecule with the anticodon UAC comes in. On the other end of our tRNA, you have the methionine amino acid. And this tRNA enters into the P site, the site in the middle. And then your tRNA binds to your mRNA. So we have our first amino acid entered in, and it's the start amino acid. Then what happens is you have elongation. So a second tRNA with a complementary anticodon comes into the A site, the first site. So if you look on this picture, so we have our start codon AUG in the P site, has the tRNA UAC, so it has the anticodon that matches. And then at the top of your tRNA, you have your first amino acid, the MET, the starting one. In the A site, you have your second tRNA come in. So our codon is CUG on our mRNA. The anticodon is GAC, and you can see that in the picture. And this tRNA holds a leucine amino acid. So we have our second amino acid come into the A site. What happens is these two amino acids that we have next to each other, a peptide bond is going to form between the two amino acids. So they're going to bond to each other. So that's the next step of the elongation process. Then your very first tRNA is going to be released, and your ribosome is going to slide down to the next codon. So, step number 4 right here, your ribosome slides down, and this is on the left, your ribosome slides down one codon. So now our second codon is in the P site, and our A site is open. So that A site's open, it allows our third tRNA to come in. So our third codon is CCG, the anticodon that matches is GGC, so that tRNA comes in, and then on the top of the tRNA you have proline, your third amino acid. So another tRNA fills the A site, and then your third and your second amino acid, you have a peptide bond formed between them. And you can see that on step number 5 on the right over here. So peptide bond number 2 holding amino acid 2 and 3 together. And then basically it just keeps on going until you get to the last part of the translation, which is termination. So this process continues until a stop codon is reached, and there's 3 different stop codons or termination codons, so UAA, UAG, and UGA. And you can find them on that chart, that genetic code chart that we looked at a few slides back. So these 3 codons, there's no corresponding tRNA to these 3 codons. So when you reach one of these 3 stop codons, instead of a tRNA entering into the A site, you have another molecule called a release factor. So when this release factor enters into the A site, that causes a ribosome to break apart, so the small subunit and large subunit break apart or they fall off, and your last tRNA is removed from the polypeptide. So termination is basically everything comes apart. So this process, it doesn't just happen slowly or just at one time. So usually on mRNA molecule, it can have multiple ribosomes following each other. And this is called a Polyribosomal Complex. And this allows for the synthesis of many protein molecules simultaneously or very fast from the same mRNA molecule. So if we look at the picture on the left, you have your DNA, the pink and the blue strand. The blue strand is our template strand, so it goes through transcription. We make a yellow mRNA molecule from it. Then, once that yellow mRNA molecule is ready, the first ribosome can attach on and start moving down your mRNA. After that first ribosome gets a few amino acids in, a second ribosome can come in and follow right behind it. So, the bottom picture, picture B, on the left, you have multiple ribosomes following each other. And then picture C, this is actually showing it from an electron micrograph using an electron microscope. So you have your mRNA molecule going through the middle of the picture, and you have multiple ribosomes attached to the mRNA molecule, one right after the other. And the last thing, so prokaryotic and eukaryotic cells, they both do transcription and translation, but they have tiny little differences between the two types of cells. So in eukaryotic cells, transcription, the first part of this process occurs in the nucleus. Then that mature mRNA travels out of the nucleus and translation occurs in the cytoplasm. So eukaryotic cells, because they have a nucleus in them, transcription occurs in the nucleus, translation occurs in the cytoplasm. And eukaryotic start codon is AUG, but it doesn't use a different form of the methionine here. And then eukaryotic mRNA encodes for a single protein, where bacteria mRNA can actually code for many proteins. And then the one big difference that I also pointed out, eukaryotic DNA contains introns, which we have to splice out. And introns are just intervening sequences of non-coding DNA, so they're extra DNA pieces. And these introns have to be spliced out to make your mature or final mRNA transcript. So that's, if you go back, that's where we talked about the introns and the exons, and how you have to splice out those introns.